Well, hey AP, hope you're doing well when you watch this video. So this is part 2 of Mitosis, about like kind of cell cycle regulation and cancer and stem cells. That's what we're going to be diving into. Full disclaimer, I've been sick for a couple of days now trying to make this video for you. So I've drank a lot of water before doing this. Hopefully I won't cough too much when trying to make it. But nonetheless, I did want to make this for you to be prepared for the upcoming quiz. Okay, so cell cycle and cell division. I'm just going to go through the entire PowerPoint again. Feel free to use this as you will. So we opened up talking about Pacific yew tree and paclitaxel, and it's a drug that comes from the tree that can treat cancer. We discussed that case study about human papilloma virus. And how HPV increases the risk of cervical cancer. And there's a case right there if you want to pause and read it. And so here's some statistics that I showed you as far as cancer goes. It's very humbling in the sense that, 1 out of 2 males and 1 out of 3 females from birth to death usually somehow, some way develop cancer. We do live in a broken, fallen world, but if you read Revelation, this world and the rulers of it and the brokenness of it doesn't have the final say. Jesus has the final say, and he wipes away every tear from every eye. There's no more pain or suffering or death. All these things are done and over with in the end. So it's it's a good thing to read Revelation, especially in hard times. And so here is the topics, the learning understandings, the objectives from AP Classroom. College Board. And so we're going to dive into checkpoints and what happens when there's disruptions. So the frequency of cell division varies with the type of cell. Some cells divide very quickly, skin, intestine, intestinal cells, hair. Some cells divide not as quick in your body. These differences result from regulation at the molecular level. So, flipping genes on or off, speeding up, slowing down, turning signals, signals can turn cell division on or off. And so cancer cells escape these controls, and we're going to dive into that towards the end of this. So the cell cycle is driven by specific signaling molecules present in the cytoplasm. So just for review, a signal is a molecule that either stimulates or inhibits a metabolic event, chemical reactions. So some evidence for this hypothesis comes from experiments with culture mammal cells. And so you can see right here that they took 2 cells and fused them together. The S cell induced the G1 cell to go into S. The M cell induced the G1 cell to go into the M phase. And so there's some sort of signaling happening. And of course, through years and years of research, we have a better understanding of what that is. And so the sequential events of the cell cycle are directed by a distinct cell cycle control system, which is similar to a washing machine. So the cell cycle control system is regulated both by internal and external controls, inside the cell and outside the cell. And so there's multiple checkpoints. And so it's kind of like a washing machine. One textbook describes it like this, and it's just like every single notch is an event, and it has to happen. Eventually there's these huge checkpoints that the cell has to make sure everything is ready and willing. DNA damage is a big warning sign, like, hey, do not pass, we gotta fix this. Chromosomes have to be attached to spindle. If that doesn't happen, we gotta stop, we gotta fix this. And so for many cells, the G1 checkpoint is called the restriction point. It's a very, very important one. Usually if a cell passes G1, it goes through the cell cycle. And so that's a very important checkpoint, okay? The cell does not receive the go-ahead signal and enters this G0 phase. Muscle cells, nerve cells in general are in that G0 phase, although there's research going into that certain times they can start dividing under certain situations. And so, let me turn this off. And so again, it's kind of like, G1 checkpoint, there's a red light there, something's damaged, we go into this halted, non-dividing state. If there's a green light, hey, pass go. Let's go ahead and go into cell division. And so cell mitosis receives a stop signal if it's not attached to spindles. Okay? But then we gotta attach and we gotta fix that. And so the cell is regulated by a set of regulatory protein and protein complexes, including kinases. We'll know these as Cyclin-Dependent Kinases, CDKs, and proteins called cyclins. Family of proteins called cyclins that increase and decrease as the cell continues. cell cycle continues. So these cyclins increase and decrease as the cell progresses through that washing machine cycle, right? Every single notch, these cyclins are changing their concentration levels. And so it's important because it allows time for any damage to be repaired. We do not want damage to be copied and replicated and passed on to the next generation of cells. This results in deformities, cancer, and we want to fix the damage. You know, of course, anytime the word damage is brought up, we want to fix the damage. And so an example of an internal signal occurs at the M checkpoint. So right, kinetochores remain unattached to spindle microtubules. Anaphase does not begin if that happens. And so attachment of all kinetochores activates a regulatory. So here's that key word again, right? It's all signaling. You don't have to know specifics for the test, right? But you have to know in general. So there's something that activates a complex which sends a signal and then activates another enzyme. This is signal transduction kind of pathway a little bit. And separates, allows this chromatids to separate. So that's just an example of how signaling works in these situations. Some external signals are growth factors. You know, we all have experienced growth, physical growth in our life. And so, for example, platelet-derived growth factor, PDGF, if it's around, the cells will divide. And so another example of an external signal is density-dependent inhibition, in which crowd cells stop dividing. So most animal cells also exhibit Anchorage dependence. So these are two external control systems, basically. Density-dependent inhibition, when they touch, they stop dividing, cells that is. And then Anchorage dependence, they have to be attached to something in order to divide. And so cancer cells exhibit neither of these, density-dependent inhibition nor Anchorage dependence. So cancer cells really have escaped some of these cell cycle control mechanisms that have been designed. So you can see here that cells, they're not anchored, they don't divide, but when they touch, they stop dividing. If you scrape some away, they divide again. So there's definitely something turning on and off cell division here. And so cells have a way, and your body has a way of protecting against damaged cells, and that's apoptosis. It's programmed cell death. We talked about in class about how you developing fingers was an example of apoptosis. Apoptosis is not necessarily a bad thing. When we hear the word death, of course we automatically go to negative consequences. But apoptosis allows development to proceed correctly, but it also allows destructive cells that have mutations or damage to be destroyed because we don't want them to continue. And so it involves a sequence of cellular events like fragmenting of the nucleus, blistering of the plasma membrane, engulfing of cell fragments. It seems pretty extreme, right? But that is the case. And so apoptosis is caused by enzymes, and mitosis and apoptosis, of course, are opposing forces. Mitosis, we're growing cell cultures. Apoptosis, we're destroying cells. So here's just kind of a picture of what apoptosis looks like. We fragment the nucleus, we blister the plasma membrane, and these fragments are eventually destroyed in the cell. So the cell cycle, cells harbor caspases that are kept in check by inhibitors. So these inhibitors, they can be activated, unleashed by signaling. And one such protein, which we discussed some in class, the guardian of the genome, P53. It stops the cell cycle at G1 when DNA is damaged. There's so many great videos out there. I know I tried to show you one the last time we met, but I would definitely look up P53, some of the videos there. It's really, really interesting. So here's a picture here of like kind of what happens in cell cycle, regulation. So if a CDK is not present, Cyclin-Dependent Kinase, that's what CDK stands for. So it's a kinase which adds a phosphate. Cyclin-dependent, which means there has to be a cyclin around for it to be activated. And then there's an RB protein. So if the RB protein's around, this E2F is not released. But however, if the RB protein is phosphorylated means adding a phosphate. CDK is phosphorylated because CDK is present. So again, it's all, dominoes, right? Step one, step two, step three. So the CDK cyclin has to be present, step one. Then the CDK is active, step two. It phosphorylates RB protein, step three. And then because phosphorylated RB protein, it releases this E2F. And E2F binds the DNA, and it allows cell cycle to progress. And so this RB protein is an inhibitor. If it's phosphorylated, it is basically shut off, and the cell cycle progresses. P53 If there's DNA damage, it's activated, and it has multiple systems to help repair proteins, and if it cannot repair, it leads the cell to apoptosis. So again, you do not have to know this feature picture here. I just want you to kind of see like these cyclins increase and decrease based upon other signals, and so the entire process is highly regulated. And so I just want to take you through it one more time, okay, just kind of from start to finish using some notes from another textbook that I have. But the restriction point, these internal signals are called Cyclin-Dependent Kinases. Okay, they add phosphates to proteins that regulate the cell cycle, therefore turning proteins on and off, and to allow the cell cycle to either happen or not happen. And so this is just control points, G1, S, G2, M, cytokinesis, and there's timing. Of course, most cells spend a lot of time in G1, S, and G2, and roughly only an hour dividing. And so most of the time is spent in interphase. And so CDKs are activated by binding to a cyclin. So it's a protein, and CDKs function at different checkpoints. You can see that DNA damage is a common triggering point, triggered by incomplete replication or DNA damage again, chromosomes that fail to attach. All these checkpoints have certain triggering points. And so once the CDK is active by having a cyclin attached to it, the cyclin it does its job, and then it's broken down by another protein. So again, this is highly regulated. So you can kind of see an example here. Here's a cyclin just in general, attached to the CDK, allows the cell to progress. That cyclin is broken down, and then it recycles all over again in time for the next round of cell division. So you can see here's that picture, concentrations of cyclins. Sorry, let me get a drink of water here. Probably should have just paused the video there. I'm not very good at making videos. I don't know all the tools of the trade. But here's that P53, just some knowledge from medicine.plus.gov about genetics. P53, it's insane how much it does. You can see right here that picture that I showed you in class about all these are situations, triggering moments, if you will, that causes P53 to be around and active. I mean, physiological processes, hormone, oxidative stress, cancer type signaling, DNA damage, low oxygen, nutrient deficiency, all of that increases P53 being around. So why is this important? Is because if we don't cancel damage in cells, if we don't stop this damage, it can lead to cancer. And one of the stories in biology that's famous story is Henrietta Lacks. I'm sure you might have heard about it. Oprah actually made a movie about her. There's a wonderful book, The Immoral Life of Henrietta Lacks. I usually have AP students read in summer work about her life and her legacy, and it's very. And so here's a timeline basically Henrietta Lacks developed cervical cancer, I believe in like the 50s or something like that. And so you can see here, she was born. Biopsy of the tumor was taken and sent to a lab, George Guy, resulting in the creation of HeLa cell lines. These cells are still alive today. She died at the age of 31 from cervical cancer. She was an African American woman. So women's rights, African rights, all this is discussed in the book. There was no informed consent when her cells were taken. It's really the book really opens up many important discussions about ethics and informed consent and patient rights and women's rights and racial issues and all sorts. It brings up wonderful talking points from a medical, ethical, but also a Christian perspective. As Christians, we're called to fight the good fight of faith. We're called to follow Jesus, and Jesus broke down barriers, and Jesus, for God so loved the world, the whole world, right? And so these are very important discussions to have. But her cells have led to development of successful drugs in fighting human diseases such as leukemia, hemophilia, herpes, human papillomavirus, Parkinson's, influenza. I mean, her cells have been used in COVID research, I'm pretty sure. I mean, there's all sorts of different things HeLa cells have been used for. What's interesting about the book is that billions and billions and billions probably have been made, through using her cells, but a lot of her family lived in poverty in the Maryland area, if you read the book. And it's a wonderful book by Rebecca Skloot, I believe. And so you can see there's a YouTube video here. If you want to click on this, the immortal cells of Henrietta Lacks to learn a little bit more. But the cell cycle in cancer, again, all of us have been affected by cancer in one way, shape, or form. I've lost loved ones and friends due to cancer, and here's the thing: cancer. When you boil it down biologically, it's a it's a growth disorder caused when cells divide uncontrollably. They're not controlled. They have no controls. That they ignore all the checkpoints. They ignore all these cell control regulations, and so it accumulates mutations that lead to loss of control. And so this leads to the formation of a tumor. Of course, it's something that you never want to hear. There's two different types of tumors. I'm sure you're familiar with. Benign are not cancerous. They do not invade. Malignant are the dangerous ones. They're cancerous. They readily invade neighboring tissues, may also detach and lodge in distant places. That's called metastasis, or metastasizes into other tissues. And development of cancer tends to be gradual. It can be very aggressive. Certain forms, of course, it's not a one size fits all. It can be, aggressive and come on pretty strong, but it's very important. So let's just look at an example from a textbook I have about breast cancer. Of course, October and breast cancer. If you scroll back, let me just scroll back up. So you can see here breast cancer. Oh, let me take this off. Breast cancer right here. 1 in 8 females birth to death. I mean, that's crazy high, right? And so screenings and physical exams, all that's super important. So you can see here breast tumor. Here's a breast cancer, colorized. Of course, colorized. Cells don't have color; they don't have pigments. And so you can see here there's a tumor, a lump, and then it can eventually metastasize. They can spread through the lymph system, which is kind of like this slower circulatory system full of like lymph nodes, and it helps the immune system kind of protect you. It can spread through the circulatory system, and then eventually it can spread to other areas, to other parts of the body. And so this is where you get the stages from. You get stage abnormal cells are there, but have not spread. So that's good to catch it early. The earlier you catch cancer, the better. Early localized, super small, 20 to 50 millimeters. Some lymph nodes are involved. Regional spread, the tumor is larger than 50 millimeters with more lymph nodes involved. This is the stage of breast cancer. And then stage 4, you never want to hear someone having stage 4. It's not automatically death sentence. The treatments have become so amazing and wonderful, and of course we praise God for all those discoveries. But stage 4 is when cancer is spread, and that's when chemotherapy and all that gets involved. And so localized tumor may be involved, treated with high energy radiation, but if the cancer spread, we gotta use chemotherapy. And so chemotherapy drugs interfere with specific steps of the cell cycle. This is why you see the side effects, like the nausea and the vomiting and the hair loss. It's because these chemotherapy drugs, they usually attack cells that divide frequently. And so you're quickly dividing cells like your intestines, your digestive system, your hair, your skin, other places. Like they destroy those cells. And so that's why you get those really bad side effects. Of course, we're trying to doctors are trying to save your life, but nonetheless, it's pretty rough. And so how does cancer cells differ than normal cells? Well, they lack differentiation, which means they're not specialized. They can enter the cell cycle repeatedly. Some cells are, immortal, like, meaning like they can divide and divide and divide, like HeLa cells. It's amazing. They still are dividing today. Are they exactly the same as Henrietta's original DNA? Definitely not. But they still are dividing. They have abnormal nuclei, could be enlarged, could be a number of chromosomes, definitely have different genes. They don't die under apoptosis. They avoid that. The immune system can recognize abnormal cells and trigger apoptosis, but these cancer cells have an ability to evade the immune the immune system. They form tumors, of course. Cancer cells, they have let me change that. They have achieve the ability to evade, these cell cycles, cell signaling regulation. So cancer cells have lost contact inhibition. Remember that, density-dependent inhibition. Like, when we're touching, cells are touching each other, they stop. Cancer cells don't care. They grow on top of one another. They can spread. They can also undergo angiogenesis, which is a crazy important word that you will need to know for the quiz. which means they literally cause the formation of new blood vessels to nourish them. So they trick your body. They send out these signals, and blood vessels literally grow towards where they're where they're growing, and they create these new blood vessels to get new nutrients to fuel their growth. So they basically hijack your system sometimes in order to steal nutrients and stuff. So oxygen and nutrients to the tumor, that's angiogenesis. Of course, genesis means beginning. So here's an important table versus, cancer cells versus normal cells. I would pause here and kind of study this, walk yourself through this. So the origin of cancer, different terms here. The first term that you need to know is an oncogene. An oncogene is a mutated gene that contributes to the development of cancer, okay? In their normal unmutated state, again, mutations, different codes, right? Four nucleotides in DNA, A, T, C's, and G's. And so mutation, meaning the code's different, are called proto-oncogenes, and they play roles in the regulation. Some oncogenes work like putting your foot down the accelerator of a car, pushing a cell to divide. Other oncogenes work like removing your foot from the brake while parked on a hill, also causing the cell divide. So here's just a little graphic again from the genome.gov website that you can see here at the bottom. Normal oncogene encourages cell growth when needed, but activating a mutation leads to accelerated cell growth, which leads to problems. There's also tumor suppressor genes, which inhibit the cell cycle in various ways. P53 is one. If a tumor suppressor gene becomes inactive, it may promote cancer development because it's not suppressing a tumor anymore. It's not stopping the cell from dividing when it needs to stop. Okay, so proto-oncogenes, activity mutation, activating mutation enables oncogene to stimulate cell proliferation, which means it's dividing a lot. If a tumor suppressor gene is mutated, two inactivations could cause the gene to no longer make the protein necessary to control cell cycle development. There's also this wonderful truth about telomeres. Telomeres are basically like shoelaces on a chromosome. If you have a shoelace, I don't know what the plastic ends are. I forgot what they're called now. Ziglets, or I forgot what they're called, but it's the plastic ends on the shoelace. It prevents it from fraying. Well, every time your cell divides, you lose a little DNA. And so literally this is in medicine textbooks all around the world, telomeres, and good telomeres, long telomeres. My dad talks about it with his doctor friends at hospice. Like, you have these telomeres, these ends called telomeres, and some of us have longer telomeres than others, and we're more able, for those who have longer telomeres, to prevent possible cancer development because we have more protective, kind of like this spare DNA at the end of chromosomes to protect us. And so when they get very short, the cell will no longer divide. And so there's an enzyme called Telomerase, which maintains the length of telomeres. And there's so much research going into this. You can click on the link here to kind of dive into it. But telomerase maintains this length. So mutation in telomerase genes cause telomeres to continue to lengthen, which allows cells to continually divide, cancer cells that is. But it also can be a good thing. Telomerase could prevent aging, and it kind of talks about that here too. So it's kind of a one in, good and bad situation. So this is just kind of a blurry picture about proto-oncogenes and tumor suppressor genes and various things like that. And here you can see just an example of how microtubules are in play and how they eventually pull cells together. But here's where our paclitaxel drug works, right? Paclitaxel actually attaches to the microtubules and destroys them. And so if you can't pull those chromosomes apart, the cell doesn't have DNA. It can't function. It dies, cell death. And so paclitaxel, that's exactly how it works. Binding to microtubules in the spindle. And so it prevents the chromosomes from separating normally and thereby kills the cell. The discovery of paclitaxel as an anti-cancer drug was based on a combination of traditional people's knowledge. So this is sometimes how medicine works. Systematic evaluation and testing using models and clinical trials. And so, yeah, there's traditional medicines out there that work. I mean, there's so many COVID-19 treatments out there, that work. And so systematic evaluation and testing using model systems and clinical trials on humans. So the Pacific yew tree grows mostly in the Pacific coastal region. So it works by binding to the microtubules and stopping them. And so they would literally test this out, and then they developed a new drug. HPV, human papilloma virus, stimulates the cell cycle when it infects the cervix. So oncogene proteins are mutated positive regulators. In cancer cells, they are overactive. And so remember, tumor suppressors are negative regulators, but they're inactive. And so for an example, RB blocks the cell cycle at the restriction point. HPV causes synthesis of the E7 protein, which fits into the protein binding site of RB, thereby inactivating it. So literally, HPV changes cell signaling, these cell signaling control points. And so this is just from a research article. You can click on the link right here. But HPVs can be divided in two groups. There's different types of HPVs. Not all are the same. Some cause warts, some are different than others. But you can see here that there's two virologies, E6 and E7, are preserved. E6 and E7 are transforming genes. It degrades tumor suppressor protein P53. There's our guardian of the genome, right? E7 inactivates the RB protein, and they disrupt cell cycle regulation. So again, right here, super important. These proteins change these two regulatory proteins that we've learned about, P53 and the RB, and HPV does that, and that's how it leads to cancer. Okay, vaccinations have been important since the discovery of this. You can get vaccinated. This German virologist, he won a Nobel Peace Prize for developing an HPV vaccine. So you can click on that to kind of read about. Henrietta has been used in there, and also some different HPV and cancer situations. So the last two slides, phases of mitosis in animal and plant cells. There's one other thing that's kind of important here, and there's stem cells. So stem cells are kind of like these undifferentiated cells. They're not blank slates, but they haven't truly developed into what they need to be. And so they retain the ability to divide. And so red bone marrow is an example. You have stem cells in your bones that produce the various types of blood cells. You have multiple different types of blood cells, red blood cells, white blood cells, and all sorts of different white blood cells. And so from this, we get into the idea of cloning. Can we produce human tissues from adult stem cells or embryonic? And so there is an ethical debate because embryonic stem cells destroy the embryo. So is that abortion? But what's wonderful I believe it was a Japanese scientist. He discovered a way to induce adult stem cells into its embryonic state. So that argument, and for most intents and purposes, it has been tabled because we don't even need to do it anymore. And so embryonic stem cells can be used for reproductive cloning, the production of a new individual. Entire herds of cattle have been cloned. You've probably eaten cloned meat. Who knows? Maybe I have too. And there's so many different links here and videos that you can watch, but here's just an example. I mean, if someone was suffering from like a cancer or heart disease or lung disease, what if we could grow them a new heart from their own DNA? What if we could scrape off a skin cell or some sort of cell and create, through signaling, a new heart that's genetically identical to them? Transplant patients, or a new kidney. Transplant patients, sometimes they have to take medicine for their lives because their body will attack that tissue. What if we could do that without? I mean, it really interesting conversations can happen from this. And so you can see G0 cells from animal to be cloned. Basically what they do is they take the nucleus out of the egg. They fuse the cell that wants to be cloned. So that's the original DNA, right? So just follow kind of the DNA here. DNA's here. Take the DNA from the mom, put the full DNA from what we're trying to clone, culture it. Of course, it's crazy complicated, involving electricity and all sorts of stuff. Sometimes it doesn't work. And implant the embryo into the surrogate mother and clone it. The first animal that was cloned was Dolly the sheep, I believe. And then you can see that the G0, sorry, that was my phone. And then therapeutic cloning, yeah. Let's remove the egg, some embryonic stem cells, and then you can see here, again, doesn't have to be, and then we develop into, any type of tissue or cell that we need possibly. So stem cells, reproductive and therapeutic cloning, very interesting. Some scientists around the world trying to bring back the woolly mammoth. Who knows? Who knows? So again, I hope this was helpful. I hope hope y'all are doing well in this crazy time of life we're living in. God is on the throne. Thank y'all so much for your time. God bless.